He got involved in a number of types of scientific activities. One that I'm proud of him for is that he got involved with a major undergraduate national journal, the Journal of Young Investigators, and he rose from, I guess, an assistant editor to editor-in-chief of that journal. Summers he spent at the Pacific Northwest National Laboratory in Richland, Washington, doing a variety of research, and he's going to talk a little bit today about his studies with C. elegans, and I noticed there he actually did work with worms, a soybean cyst nematode, which causes about $500 million of damage to soybean crops in the US each year. Also got involved in protein folding. But one of the things he then got involved in is research with Utpal Banerjee and John Olson. And here he learned collaborative research. And he ended up on 2 research papers. One of them was in the journal Genetics that had literally a cast of hundreds, okay? And then another paper in Nature Methods, and that only had a cast of tens. But I think from this, Alexander learned to work with lots of people. Alexander has lots of honors. You know, for someone finishing a PhD to have sort of an 8-page CV, you know, in sort of, you know, 7-point font is pretty impressive. And so Alexander has a whole slew of honors, including the John M. Jordan Award from the Department of Chemistry and Biochemistry for Creative Studies, the Audrey Fowler Award in Protein Science, a Dissertation Year Fellowship. Alexander loves to go to meetings and talk, actually 6 invited meetings, including the 25th International Conference on Yeast and Yeast Genetics and Biology, Molecular Biology in Poland, and Alexander's abstract was selected for a talk there. He's actually given 6 invited talks so far, and 27 poster presentations, okay? So this is a guy that loves to talk, and he loves to go to meetings, and in fact, you know, Alexander, he doesn't care if it's the UCLA Sigmund Symposium or the UCLA Seaborg Symposium or the MBI Lake Arrowhead Retreat, or even better, going off around the country. So I just looked at some of the places he given posters. So national, international meetings, San Diego, Washington, D.C., Charlotte, North Carolina, Bethesda, Maryland, Boston, Massachusetts, and Vancouver, Canada. One last thing I'll say before I let him go is that Alexander has been a fantastic person to have in the lab. He's been a fantastic mentor. I saw 4 undergraduates that Alexander has mentored long-term, 2 post-baccalaureate students long-term mentorship. He's had 6 rotation students under his wing. He's also been selected to be a UCLA graduate research mentor for undergraduates and is involved in those activities. And lastly, has been sort of the scientist in the lab, that he's somebody you can go to for advice on anything. He has an amazing ability with complex instrumentation and amazing ability with computers. So Alexander is going to weave together a number of stories today, and I will let him go without taking too much more of his time.
Thank you, and today I want to talk to you about the past 5 or so years I've spent in Dr. Clark's lab. But before I begin, I'd like to say thank you to Dr. Clark for being such an amazing mentor. I'd also like to thank Dr. Jonathan Lowenson, which is somewhere out there, over there, who has been extremely helpful as well during the past several years, as well as everyone in the lab. I couldn't have reached this point without everyone's help. I'd like to start by making an analogy and comparing a cell to a car. Now, like a car, cells have proteins that make the cell function. Some of these proteins are essential, like the spark plugs of a car. Some of them are not essential at some points, but in the dead of the night, they may be, such as the lights. And some degrade over time, such as the wheels. Now, if you dissect the car, and don't get into a battle about different models, but a typical car has about 15,000 to 20,000 different parts. Now, how does that compare to an actual living cell? Well, if you look at the genome, there's about 20,000 different genes in the mammalian genome. Now, one would expect that, okay, you have 20,000 different genes, you'd have 20,000 different proteins. Well, that doesn't appear to be the case. In fact, if you look at the transcriptome, you can read genes in different ways, such as a computer program you can read in different ways. And instead of 20,000 different transcripts, you can have 100,000 or more different transcripts. Now, that's a lot of diversity. But once you make a protein, that diversity goes up even more. So instead of having 20,000 different proteins or 100,000 different proteins, you can have 2 or 3 orders of magnitude more protein than what the genome encodes. And that's due to what we call post-translational modifications. Now, thus far, we know that there's about 200 different types of PTMs, or post-translational modifications. And they range from very small modifications, such as hydroxylation events, to very large modifications, such as the addition of proteins to other proteins. Today, I want to talk about really 2 types of modifications. One that's called methylation, and another which is called isosporyl formation. Now, to understand methylation, one must understand how does that actually form. Well, a substrate and S-adenosylmethionine are recognized by what we call methyltransferases. And the methyltransferase will transfer that methyl group from S-adenosylmethionine, which I'll call 8-O-met, to the protein substrate, forming a methylated substrate, and S-adenosylhomocysteine as a product. Now, what this methylation does is amazing, and you can't really look at an area of biology without seeing the importance of methylation. But for basics, methylation regulates transcription and translation, is involved in repair of not only DNA, but also proteins, and has been linked to actually even viral infections. But I wanted to learn during my 5 years in the Dr. Clark lab about what more we can learn about methyltransferases. Are they static entities, or can they interact with one another to change global methylation? Do they always methylate, or can they do other unique things? Are they always important, or can they have compensatory pathways? And can just studying a methyltransferase lead to the discovery of new biology? So we're going to start my journey here through the past 5 years, and we're going to start off with arginine methylation. Now,
should we have less lights in front?
Yeah, that would be great.
Maybe. Can somebody maybe get lights in the back? I'll turn these off. Oh,
No,
is that good?
that's
Was that
not
better?
much.
Was that even better?
Yeah, I think
No,
Okay.
this is fine.
So let's start off with arginine methylation, and we're going to start with arginine methylation because it is one of the most prominent types of protein methylation in the cell. Now, arginine can be methylated in 3 different ways, and that's methylated by 3 different methyltransferases. First, you have type 1 arginine methyltransferases, which can methylate arginine once to form monomethylarginine, or MMA. It can then methylate it again to form asymmetric dimethylarginine, or ADMA. Type 2 methyltransferases also form MMA, but they take it a step further, and instead of methylating the same nitrogen, they methylate a different nitrogen and form symmetric dimethylarginine, or SDMA. Type 3 methyltransferases just stop at forming MMA. Now, one of the most prolific arginine methyltransferases in the cell is PRMT1, which accounts for 85% of arginine methylation in mammalian cells. Now, PRMT1 is a type 1 methyltransferase, which forms MMA and also forms ADMA. Now, what hadn't been studied up until this point was what happens if you take away PRMT1? It is so prolific in its methylation, what happens to methylation inside the cell without PRMT1? And that's what I wanted to test. I hypothesized if PRMT1 is removed from a mammalian cell, that you would lose its products, MMA and ADMA. And that SDMA probably wouldn't change too much because PRMT1 is not involved. Now, how does one actually identify methylated substrate? Well, in our lab, we like to deal with radioactivity. And we like to make S-adenosylmethionine radioactive. In particular, the carbon or hydrogens of that methyl group. And what we like to do is incubate that radioactive 8-O-met with substrate, either a pure protein, extract, small molecules, you name it, in the presence of a methyltransferase, which will then methylate a substrate. What we then do is we separate that sample. For example, if we will look at protein methylation, we may acid hydrolyze the protein into amino acids and separate that on amino acid column. We can also use high-performance liquid chromatography to separate peptides and proteins, and we can also use SDS-PAGE to separate out these species and put radioactive film on top to see which protein bands are radioactive. This is all well and good, and we've done this several times. But then the question is, how can you do the quantification of arginine methylation in an intact cell? Now, if we do yeast, that's relatively easy. You take the yeast cell, you put it with Adomet, you incubate it, and then you analyze the extract. But in mammalian cells, it's a little bit more difficult because they can't really import Adomet well. And if we want to look at biologically relevant modifications, we need to come up with another way that isn't radioactive. Now, teaming up with our collaborator Mark Bedford at the MD Anderson Cancer Center, we looked at that. And what I did was I obtained wild type and PRMT1 knockout mouse embryo fibroblasts from the Bedford lab. And then I acid hydrolyzed the entire cell, converting the proteins into amino acids. I then separated out all these amino acids on a cation exchange column. This is how these arginine derivatives look like on a cation exchange column. These are just standards. If it was a biological sample, you actually would not see anything. And in this case, we're actually looking at a color change due to the chemical anhydrine. Now, you may notice that we can separate ADMA, SDMA, but we can't separate arginine from NMA. So the separation is not complete. We need a second dimension. And that second dimension comes from reverse phase Hplc. And not just any ordinary reverse phase Hplc. To quantify now what we have, we derivatize the fractions we obtain from cation exchange with this molecule called O-phthalodialdehyde. This reacts with free amines of amino acids, forming a fluorescent molecule, which we can quantify. This is an example of how we can separate different standards of MMA and ADMA and ADMA and SDMA. Now, if you actually look at the biology, something amazing happens. Here we're actually looking at the number of modified residues per 1,000 arginine in the cell. And we look at the wild type mouse embryo fibroblasts, and we can see that they have certain ratios of NMA, ADMA, and SDMA. But if you look at the wild type cell, something interesting is apparent. First, unsurprisingly, ADMA levels drop. They're about a half in the knockout cell. That's what we expected. But if you look at the MMA and SDMA levels, they actually go up, almost 5-fold and 3-fold. Now, what's going on? Well, we think there's an interaction going on with PRMT1 and other arginine methyltransferases. PRMT1 may sit on the substrates of these other methyltransferases, sequestering them, blocking them from methylating substrates as well. But in the absence of PRMT1, these other methyltransferases are set free, and they can then modify these different substrates. What these methyltransferases are, we don't know, and that's an area of ongoing research. That's step one, looking at PRMT1. But what about looking at how methylation changes when we analyze a different methyltransferase? And that road is going to take us to a protein called LAA, or loss of AFLR expression A. LAA is a very interesting protein that has a sequence very similar to arginine methyltransferase. Now, to understand why we even look at LAA, we must understand why it's important. Now, LAA is found in the fungus Aspergillus. And if you've ever done E. coli plates or yeast plates, you may have seen this contamination growing on it. And that was thanks to Aspergillus. You can thank that fungus for that. Now, Aspergillus is particularly bad because it's everywhere. In fact, right now, you're breathing in its spores. That's how the spores look like. If you go home to your bathroom, you may see mold on the walls. That's Aspergillus. Now, it's the most common component of black mold, and it's everywhere. And those that are immunocompromised, this is particularly bad because they breathe in the spores grow in the lungs, and they spread throughout the body. And thus far, we know of no known cure for it, and usually it's fatal. Now, if that's not bad enough, a fungus growing inside of you and killing you, Aspergillus also produces secondary metabolites, molecules that aren't really necessary for survival, but things that it produces. And Aspergillus produces aflatoxin and spermatocysteine as secondary metabolites. These are the most carcinogenic compounds we know. In fact, if you have a low dose of them over time, you'll get liver cancer. And if you have high doses of them all at once, you die. So it's a serious problem. Now, that is not the end of the story in terms of secondary metabolites. In fact, Aspergillus produces good things. Penicillin, lovastatin. It's actually used to make sake, as well as miso soup. And it also produces pigments by secondary metabolites. Now, what is particularly interesting is that LAA is the master regulator of all secondary metabolism and of this invasiveness in organisms. LAA, again, stands for loss of AFLR expression A, and usually LAA is found in the cytosol. But when it wants to produce secondary metabolites, it goes into the nucleus, with its partners, VA and VLB. We don't know what they actually do yet. And it interacts with the transcription factor AFLR to lead to the transcription of genes and proteins necessary for the second metabolism. Now, when you remove LAA, the fungus is no longer virulent, it doesn't produce second metabolites, and it loses pigments. And unfortunately, when you overexpress LAA, bad things happen. You get a more virulent strain, you get more second metabolites. Now, I am, again, interested in this because LAA, if you look at its sequence, contains motifs that are common to methyltransferases. In fact, if you look in the literature at LAA, people will reference LAA as an arginine methyltransferase, and that it can methylate histones, even though there's really no evidence for such an activity. No one has actually looked at LAA's activity experimentally. Now, what I wanted to figure out is LAA's substrate, because if I could figure out its substrate, I could solve this problem of the fungus. Now, what better way to start than confirming if LAA is a histone or arginine methyltransferase? Now, to do that, we come back to PRMT1. And PRMT1 is a great control because it's an arginine methyltransferase and it's a histone methyltransferase. And what we do is we combine histones, for example, in the first case, with radioactive Adomet, separate it on a SDS page gel, then put the film on top of that gel to see where the radioactive bands are. In that control, we see nothing because there's no methyltransferase. Next, we add histones and PRMT1 and radioactive Adomet together, separated by Kumasi, put the film on, and we see that the histones are methylated. Now, we can do the same thing with LAA. Combine histones, LAA, and radioactive Adomet. And what do we find? We don't find histone methylation, but we find one band, and that band corresponds to LAA automethylation. Now, LAA is rather insoluble by itself, so we have to make different constructs. That's why we have MVP LAA. But we wanted to make an independent experiment of this, and we said, okay, if we truncate the N-terminus of LAA, make another soluble version, will it also automethylate? And what we found was doing the same experiment, there was no histone methylation, but again, we saw automethylation. Now, this was very perplexing, and I spent 3 years trying to find its substrate using in vivo techniques and also in vitro techniques, looking at the nucleus, looking at cytosolic extracts, looking at small molecule extracts. But I couldn't find anything, except every time I did an assay, LAA was automethylated. So at one point I just said, okay, we're just going to determine the automethylation site of LAA, because maybe that site is important for its function. In fact, it may actually point to its methylation on substrates. Arginine methyltransferases, for example, sometimes automethylate themselves, and they automethylate at an arginine residue. So what the plan was to replace the site of automethylation of LAA, if I found one, with a non-methylatable residue, put that back in the fungus, and see does the second metabolism go away. Now that was done, so what I did was that, for example, I cut out SDS page bands where there was automethylated LAA, I acid hydrolyzed the protein, separated that on the cation exchange column. This is how that data looks like. In red is the radioactivity, resembling any amino acid that would be labeled. In black is the anhydride. This marks the standards. In every sample, we add non-radioactive standard to see where things run. Now, what you can see is that the radioactivity is right here at about 45 minutes. It is not where arginine methylation is, and it is not where lysine methylation is. And we really never see anything run in this particular location. So could this be a new type of modification? Well, I went through our inventory and I found every methylated amino acid we have. And I said, okay, I'm going to separate all these different amino acids on the cation exchange column, see which one elutes in this vicinity. None of them eluded at that vicinity except for one. And that one was a methionine that was methylated, or S-methylmethionine. And that eluded with our sample. Now, we wanted to confirm this, and we said, okay, we're going to mutate every single methionine residue in LAA. And my collaborator, Jonathan Palmer at the University of Wisconsin, did that. He mutated every methionine. There's 5 different methionines in LAA. And then I incubated that with radioactive Adomet, ran it on the cation exchange to see if automethylation was still present. In every case, automethylation was present, except for one. And that was when methionine 207 was converted to alanine. and we saw automethylation completely removed. Now that brought up the question, why is this residue, methionine 207, methylated? Now one could go and they could look at the crystal structure of lay to see why. But unfortunately, lay is a very difficult protein to handle, and I tried to crystallize it, and several people around the world have tried to crystallize it without any real success. So what I had to resort to is three-dimensional modeling using the program FIRE2. I modeled the structure of lay A, and I found the methionine 207 residue. Now what FIRE2 did was then it inputted Adomet. I didn't tell it to input it, it inputted it itself, and then it put it in this particular location. And if you can see, Adomet is basically right next to methionine 207. In fact, if you look at the reactive sulfur group of methionine 207, and you calculate its distance to the transferable methyl group of Adomet, the distance is 5 to 7 angstroms away. It's basically asking to methylate. Now with such an interesting combination, one must think this must be biologically important. Why would you put radioactive groups next to each other? And so what my collaborator did was he looked at that. He looked at first the pigmentation and also secondary metabolism, most specifically stegmaticystin production by TLC at the bottom. In wild type Aspergillus, you can see the colonies are usually green at the top and kind of brown at the bottom, and they produce secondary metabolites, which is shown here on the TLC. When you mutate lay in Aspergillus, you see that the colonies turn white, and then you lose secondary metabolism. Now we have this knockout strain. What happens if you put back in lay A? Well, if you put in wild type lay A, you see that it restores pigmentation and it restores secondary metabolism. And unfortunately, when you put back lay A that has that methionine mutated to alanine, you have a wild type phenotype. It rescued it. It suggested that it actually wasn't biologically important at this point. We took this a step further and we said, okay, maybe Adomet must bind to lay A to get automethylation. So what happens if we mutate the Adomet binding domains of lay A? How would that change secondary metabolism? That was done, and you can see here that when you mutated various residues involved in binding Adomet, that you lost pigmentation and you lost secondary metabolism. Now what does this all mean? Well, methionine 207 in filamentous fungi is conserved. It's not found in one filamentous fungus, and in that fungus, it does not produce secondary metabolism. So it appears to be important. Now, if you cannot bind Adomet, I found that lay A doesn't automethylate. And what my collaborator found is that it can't dimerize, and it can't go into the nucleus. So that all suggests, okay, they can't produce secondary metabolites, so maybe lay A has some sort of novel function. And what I believe is going on is that lay A is actually not maybe a methyltransferase, but actually a sensor of Adomet levels in the cell. For example, if Adomet is not present, lay A can't bind it, it's cytosolic, and you get no secondary metabolism. But if Adomet is present, lay A will bind it, dimerize, go into the nucleus, and then you have secondary metabolites. The function of methionine 207 there could be many, but one could be to adjust the sensitivity of this sensor. Okay, thus far we've talked about kind of regulatory methyltransferases, PRMT1 and lay A. But what about talking about more of a methyltransferase that responds to spontaneous damage, more of a repair methyltransferase? And that's going to lead us off to another path, and that path involves isospardal damage. Now, to understand this path, one must understand what isospardal damage is. Isospardal damage arises from two amino acids that should never have been put in the 20 amino acid code. And those are asparagine and aspartame. These amino acids are particularly bad because the backbone nitrogen can attack the carbonyl group of these residues. When that happens, the deamidation and isomerization result in the formation of a succinimidal ring, which can non-enzymatically hydrolyze into either the spardal residue, which is not so bad. But unfortunately, that's not the primary product. What the primary product is an isospardal residue. Now, this isomerization to an isospardal, or deamidation, results in a kinked protein, because now you put the side chain into the protein backbone. And a good example of what happens to a protein when an isospardal forms is found in the crystal structure of RNase U2B. In green is the wild type structure, a portion of that protein. And I want you to look at asparagine 32. Because when you get an isospiral at this site, you see now that we have a loop that moves. Now that loop can cause many different problems. And we have linked it to aging, to the decrease in efficacy of pharmaceutical drugs, particularly peptide and protein-based drugs, and also to autoimmunity disease, because a protein that was native is now looking foreign. Now, fortunately, most organisms have a way to repair this damage, and that way is the protein isospiral methyltransferase, or PCMT. Now, what PCMT does is it recognizes isospiral damage in the cell. It binds adenine, and it methylates it, forming a methyl ester. This methyl ester will then break down, releasing methanol, to get you back to the succinimidal intermediate. The enzyme will not let go until everything has been repaired to the spiral group, because once you're at succinimide, you have a choice of going either way. Now, the importance of PCMT can be found in many organisms, but I think the best example of PCMT is in mice. Now, if you look at a mouse that is wild type or has a heterozygous mutant for PCMT, you see that they accumulate isospiral damage up to a point, and then it plateaus throughout life. And it basically has a way of controlling the damage. Now, if you have a homozygous mutant for PCMT, you see the damage levels increase exponentially, and then they stop. And they don't stop because I was cutting it close to this presentation and I didn't put the data. They stop because the mice die of epileptic seizures at about 42 days of age. Now, what's further interesting is if you analyze these mice, you find that in their urine, they actually have an increase in isospiral damage, suggesting the mouse is trying to save itself by some other pathway, potentially degrading those damaged proteins into peptides. Now, what are these isospiral Canadian species? We know there's isospiral damaged peptides and proteins. Now, what I wanted to do is figure that out. And so in collaboration with Dr. Sunny Zhou at Northeastern University and Barry Karger, what I did was I fractionated wild type and PCMT deficient urine into a peptide fraction, which was everything less than 10,000 kilodaltons in size, and then into a protein fraction, everything greater than 10,000 kilodaltons in size. I then did a traditional biochemistry approach in which I used recombinant human PCMT and radioactive adenine. And I labeled all the isospirals in yeast in the urine and separated it out on an STS page. Put film on that, and I found all the radioactive bands that correspond to isospirals. My collaborators, on the other hand, used the same sample and did a mass spectrometry approach to identify the proteins. Now, this is what I found in wild-type cells. There's not much going on. In fact, you look at the proteins on the Kumasi, and here in the fluorograph, which identifies isospiral damage, we don't see much. We see this band going across, and that's automethylation of PCMT. In the knockout cells, however, we see that isospirals are present. So we have a lot of proteins in the Kumasi, and then a lot of isospirals in the fluorograph. Now, what are these? Well, our collaborators found that these proteins resemble major urinary proteins, or MUPs. And they found several different varieties of MUPs in the urine, and they found also that they had an increased chance of forming an isospiral compared to mice with the repair enzyme. Now, this all suggested that isospiral damaged proteins and peptides can be repaired by PCMT, but they can also be degraded by some mechanism which we didn't know about. Now, up until this point, we knew about 3 different enzymes that could degrade isospirals. But in all these cases, they could only break down di and tripeptides. Now, what I wanted to do is find an organism that doesn't have PCMT and find out how it repairs this damage, because it's a ubiquitous type of damage. To do that, I took the human PCMT protein, and I looked at many different organisms and compared its sequence to those. And this is kind of a phylogenetic tree in which we find the best matches at the top and the worst matches at the bottom. Now, as you can see, animals have PCMT, plants have PCMT, fungi have PCMT, bacteria have PCMT. And this is based on the phylogenetic tree as well as actual experimental data. But interestingly, organisms that don't have PCMT kind of cluster at the bottom, and they include non-seed-producing plants, some protists, and interestingly, the budding yeast, Saccharomyces cerevisiae. Now, Saccharomyces cerevisiae is such a commonly used model organism we use in our lab. So what I wanted to know is, okay, yeast don't have PCMT. Do they therefore have high isosporal damage? Because if they didn't have any way of repairing it, they probably had pretty high levels. So I quantified the levels of isosporals in yeast. And I did that for different periods of time. So I inoculated cultures and then looked at the yeast 1, 3, or 7 days after inoculation. And I used different media, rich media, YPD, or more synthetic media. And I found damage levels range between the high 100s to high 200s in terms of picomoles of isosporals per milligram of total protein analyzed. Now, these are just numbers. How does that compare to other organisms with PCMT? Well, if we look at E. coli, wild-type cells have about 450 picomoles of damage per milligram of total protein. And if you remove PCMT significantly, you do the same thing to worms, and it also goes up. In mice, in brain, you have about the same level as in yeast, but again, if you mutate PCMT, levels go up. This suggests that some active mechanism is in play to remove isosporals in yeast. So what I originally thought was, okay, maybe common degradation pathways in yeast are degrading the isosporals. Could be the proteasome system, which selectively degrades proteins, or it could be autophagy, in which we have bulk removal of damage. Now, I tested these two pathways by inhibition and mutagenesis, and I don't expect you to actually look at all this data, because there's a lot of different conditions tested. But in every condition I looked at, whether or not it was using inhibitors or mutations of the proteasome and autophagy pathways, there was no difference between those and controls, suggesting that these pathways were not significantly involved. Now, then I kind of hit a detour, and I wasn't going anywhere anytime soon until I started doing these experiments called in vitro aging experiments. And these experiments involve taking yeast extract, adding some buffer to it, assaying them over a period of time, incubating them at a certain temperature, and looking how isosporals increase over time. Now, what I found was that in mice and E. coli, they have a certain amount of damage to start off with, and they have a certain rate of isosporal formation. But if you look at yeast, they have a slower rate of formation and a lesser total of isosporal damage. Now, this was interesting because it suggested something was active in those extracts to block or prevent isosporals from accumulating too fast. So I said, okay, can I add inhibitors, protease inhibitors, for example, to these extracts, in vitro age them, and see how the rate of isosporal formation is changed? I did that, and I used protease inhibitors such as Roche Complete protease inhibitor tablets, or soybean trypsin inhibitor, or lupeptin, A-protein, PMSF, and EDTA. And there's a lot of lines here. But what I want to point out is here's the control, basically yeast incubated in tris and aged. But then you see that there's kind of two different populations. There's 3 lines here, and the rest are here. What this appears to be is that these lines correspond to samples incubated without EDTA, a metal chelator, and these 3 lines correspond to samples with EDTA, suggesting that maybe a metalloprotease is degrading the damage. Now, we looked at this damage by SDS Page, and what I did is I just looked at the tris and the tris plus EDTA samples over 0, 2, 4, and 6 days. After those periods of time, I added the recombinant human methyltransferase, Adomet, assayed them by gel, put film on top, and every band you see here is an isosporal damaged species. What you can see here is in the tris buffer, you get an increase in damage over time. But if you add EDTA, you accumulate isosporal damage at the low molecular weight, suggesting peptides are accumulating and aren't being degraded. Now, where do these isosporal peptides come from? Well, I collaborated with Dr. Julian Whiteleg and Joseph Capri here at UCLA, and I gave them the sample, and they analyzed them by mass spec. And what was interesting is that we found several peptides that contain isosporal. We didn't find any isosporal-containing peptides in extracts incubated without EDTA, but only in samples with EDTA would we find isosporal damaged species. What's further interesting is when I analyzed the peptides, all the peptides in our samples that did not contain isosporals, I analyzed the first 4 amino acids at the N terminus and the last 4 at the C terminus, I saw that they're a mixture of everything. But if I analyzed the isosporal containing peptides, I saw that the C terminus, again, had a mixture of everything. But at the N terminus, about half the time you'd have a deaminated asparagine, suggesting that there's something specific that's recognizing that isosporal. Now that led me to conclude 2 things, or give 2 hypotheses of what's going on. And both involve metalloproteases. The first hypothesis is that metalloproteases are preventing isosporals from even forming. The logic behind that is that isosporals take, at best, 1 day to form. Now, if I looked at the peptides that I have found that contain isosporals, the average peptide belonged to a protein with an average half-life of 85 minutes. Okay, so that's clearly not 1 day. And in fact, if you analyze all the proteins in yeast, the average half-life of a protein is 43 minutes. So what it appears that maybe metalloproteases are just cleaving up proteins before they even have a chance to form as an isosporal. An alternative hypothesis is that there's an isosporal-specific protease that is finding the damage and cleaving. And that's supported by our sequence analysis. Thus far, we know that there's about 47 different metalloproteases in yeast. And right now, I have a talented rotation student, Rebecca Warmack, who's now looking at yeast strains deficient of all these 47 different metalloproteases, searching for if we can find that one specific one. Okay, thus far, I've talked about regulatory methyltransferases, PRM2-1 and LAA. I've talked about repair methyltransferases, PCMT. But now, with the remaining time I have, let's have a little bit of fun, and let's look at 2 cases of serendipity. Now, if you don't know what serendipity is, basically serendipity is sheer luck. It's basically finding something you didn't set out to find in the first place, and then stumbling upon it. And the first case of serendipity I want to talk about involves ascorbate. Now, previously, researchers in the lab wanted to know the significance of the isosporal methyltransferase in C. elegans. And that methyltransferase is called PCM1 in C. elegans. And so they analyzed the gene structure of PCM1. What they interestingly found was if they looked at the opposite DNA strand, there was another gene, known as C10F3.4. Now that gene, interestingly, had an exon that overlapped PCM1. And it just so happens that there's only 5% of overlapping genes in C. elegans that have an overlapping exon. Now this was important, and what they found was that if they searched for a homologue of C10F3.4 in other organisms, they found that it was similar to the plant VTC2. This spawned other research, which eventually found that VTC2 was the last step in the vitamin C pathway. It was the mystery step. So by sheer luck, we found that. Now that brought up the question, okay, what organisms have vitamin C? Well, rats have vitamin C, all photosynthetic organisms to date have vitamin C, some birds and bats have vitamin C. Interestingly, you and I don't have vitamin C, which is also known as ascorbate. Some birds and bats don't have it. Guinea pigs and some primates also don't have it. But it's been unclear if invertebrates do not or do have it. Previous research by others have suggested there's ascorbate in C. elegans. But what I wanted to do was prove that it was there and prove that it was synthesized. Now, I confirmed the presence of ascorbate in C. elegans extracts by 2 independent methods. One was using high-performance liquid chromatography, in which I separated out all the extract of C. elegans. And what I found was there's a peak that eluted at a position where my ascorbate standard would elude. I then combined that extract with ascorbate oxidase, or AAO. And that's an enzyme that degrades ascorbate. And I saw that peak went away. So that suggests that was ascorbate. I used then another method. I separated out C. elegans extract by gas chromatography mass spectrometry. And then I searched for an extracted ion current of 332 M/Z, which is kind of a fingerprint for ascorbate. And I found there was that fingerprint. I looked further at the fragmentation pattern of that species and it matched ascorbate. So we know now that C. elegans extract has ascorbate. And not to bore you with the details, I found that this ascorbate was not an artifact. It wasn't because of the buffer or because of the food source E. coli. I found that that gene that started off this whole project, C10F3.4, was actually not involved in ascorbate synthesis. You could delete it, you'd still get the same level. C. elegans has many homologs to known ascorbate pathways, but interestingly, it's missing some of the key enzymes. And it suggests that it may have a new pathway. So what I wanted to do is just confirm that C. elegans can synthesize ascorbate. Now what I did was I did two different conditions. I fed C. elegans unlabeled E. coli, and I let them incubate for a certain amount of time. I then lysed the C. elegans, analyzed them by gas chromatography and mass spectrometry, and searched for this particular fragment, 449, which corresponds to a derivatized ascorbate, which contains all 6 carbons. I then did another experiment where I fed E. coli C13 labeled glucose, making it heavy. I then fed that to C. elegans. Again, I incubated that for a certain period of time, extracted the small molecules, analyzed it by GCMS. If ascorbate is being synthesized, it would pick up the heavy label, and because there are 6 carbons, it would be 6 units heavier. So instead of 449, it would be 455. Now this is what I found. In the unlabeled E. coli, in the C. elegans fed unlabeled E. coli, I found the 449 peak. And when I added the heavy label, I found that 449 peak, and I also found the 455. And that suggests that ascorbate was being made in C. elegans. Finally, I wanted to see how does ascorbate level change during the life cycle of C. elegans. Now, C. elegans starts off as an egg, and it hatches and it forms an L1 larvae. And that L1 larvae has about two weeks to find food, otherwise it dies. If it does find food, it can then proceed to the L2, L3, L4, and become an adult again so that the cycle can repeat. What I wanted to do is find what the concentrations of ascorbate were in the egg, the L1 larvae, and then in mixed populations containing everything. And I did this work also with another rotation student, Charles Wang. Now what I found was that ascorbate levels in the egg of C. elegans were the highest, at about 180 micromolar. Levels then seemed to drop off as we looked at the L1 larvae. But then in mixed populations, we saw higher levels, which I believe is just because we had eggs in that population. In comparison to other organisms, we see that ascorbate levels in C. elegans are really low, even in comparison to Saccharomyces cerevisiae, which actually doesn't have ascorbate, but its cousin erythroascorbate. Now what this all suggests is that ascorbate is being synthesized by vitamin C. Ascorbate is being synthesized by C. elegans. And this is the first evidence of an invertebrate synthesizing this molecule. What I believe is that this synthesis is not due to a known pathway, but rather due to a novel pathway that may be present only in invertebrates. Ascorbate is highest in the egg stage and may be so to protect it from environmental damage or because it's involved in enzymes required for making the shell of the egg. That's the first case of serendipity, ascorbate. But there's one more thing that I want to end off with, and that's ethanol. Now we go back to the life cycle of the worm, and you start off with the egg, and the egg hatches and forms an L1 larvae. Now that L1 larvae, as I said before, has a certain period of time to find food. It's stress resistant, it needs to find food so it can live on. Now what people in the lab previously wanted to do was to look at PCM1, that isosporal methyltransferase mutant worm, and see if that L1 larvae could survive better or less than wild type under certain conditions. One of the conditions they looked at was incubating L1 larvae in minimal media called M9 media. It's basically just salt water. And they found that wild type and PCM knockout worms lived about the same. Interestingly, they added cholesterol to this M9 media, and they found the worms lived about twice as long. There's another media that you can use called S medium, which naturally contains cholesterol, and that will also produce a longevity effect. Now this was published, but interestingly, when it was published, a reviewer commented, okay, but did you do the cholesterol control? What was cholesterol dissolved in? And what cholesterol was dissolved in was ethanol, a common solvent. So this spawned another paper by another student in the lab who looked again at L1 larvae incubated in enzyme media and in cholesterol, and they reproduced the data. As you